All right, so finally we return to inflammation now. And we're going to talk about specifically the part of inflammation where neutrophils are in the bloodstream and they leave tissue because they're attracted to a site of injury. So how do they do this? Well, there are four main steps. And the first one is rolling. So before the neutrophils can even get into this tissue, they have to sort of roll along and loosely bind to the endothelium. So there are certain receptors that actually mediate the rolling step. Do you know what those are? So they're actually selectins, E&P selectins, so that you can think of them selecting for the neutrophils before they're tightly bound. And there are actually corresponding receptors on the neutrophils. Do you know what those are? Sialyl-Lewis X receptors. So both of these two receptors on the endothelium and the neutrophils, they bind to each other and facilitate the rolling step. Okay, so that's good. We've got this little neutrophil rolling along, but it's not tightly bound yet. So that's the next step, is tight binding. And just like there are receptors for rolling, there are receptors for tight binding. What do you think they are on the endothelial cells? Intercellular adhesion molecule 1, or ICAM for short. You just got to remember these. It's a little bit hard to remember, but try. And how about the neutrophil? Very good. LFA1, or leukocyte function associated antigen 1. Both of those receptors mediate tight binding. And here's a little integration here. So why are these endothelial cells and the neutrophils presenting these types of receptors? What sort of cytokines are stimulating them? So how about for the endothelial cells first? What do you think? The answer is interleukin-1 and tumor necrosis factor. How about the neutrophils? C5a and leukotriene B4. C5a, remember, was a part of what pathway? Complement. Continuing on with extravasation. So that's very good. We have the neutrophils tightly bound now. So now we need to get them into tissue. How are we going to do this? How are they going to dialyze through the endothelium? Well, the next step is diapedesis. And you can think of this as neutrophils peeking through the endothelial cells because that's actually the receptor that is responsible for this step. So the neutrophils peek through by binding to the PCAM receptors. All right, great. So we've got neutrophils in tissue. Now what? Now they just need to get to the site of injury. And that step is migration. And there are actually several cytokines, chemotactic cytokines, that help beckon or call the neutrophils close to the site of injury. Do you know what those cytokines are? There's actually a very helpful mnemonic that is called CILK, silk. So think of neutrophils being as soft as silk. For some reason, that it's kind of silly, but it will help you remember what draws neutrophils close to injury. So the C stands for, do you know? C5a. So the same thing that helps stimulate the synthesis of receptors on neutrophils actually is responsible for chemotaxis as well. So C5a from complement, the I for interleukin-8. Remember, there's a cleanup on IL-8. The L, I'll give you a hint. It's another thing that actually helped the neutrophils make those receptors to stick to the endothelium. Leukotriene B4, very good. And the K? Kallikrein. Very good. So that is how neutrophils leave the bloodstream and become attracted to injury in tissue. All right, so now it's time to talk about a very important subject, and that is free radicals. So, first of all, what are they? Free radicals are essentially good molecules gone bad. So good molecules like oxygen and water, they've undergone some transformation that left them with an unpaired electron in their outer orbit. This unpaired electron is extremely volatile and very reactive, even with normal substances in your body, like the cell membranes. Oh no. So if they react with cell membranes and damage them, then that is definitely no good and will definitely lead to a lot of cell injury and destruction in your bodies. So that's why they're so important. Where do they come from, first of all? Where do they form? Well, did you actually know, like, for example, the superoxide free radical actually forms within your body. What? What type of cell would be so malicious to do such a thing? Well, actually, the neutrophils. Remember how we talked about how neutrophils combat pathogens like bacteria and viruses? Well, they use those reactive oxygen species, remember? And that's what we're talking about. Free radicals are reactive oxygen species. So they will use them actually to kill pathogens, but they've got to be controlled or they can actually harm your own cells too. As we saw with, remember when we saw that? Reperfusion injury, remember? Yeah. So what enzyme actually is responsible for generating this superoxide free radical? It's actually NADPH oxidase, also known as the respiratory burst. Very good. There's actually another enzyme possessed by neutrophils that can generate another free radical from this superoxide. What is that free radical, first of all? Peroxide. And what's the enzyme? So it's actually superoxide dismutase. So it's dismutating this superoxide free radical. Very good. And in turn, this peroxide can actually turn into another type of free radical that is most harmful to pathogens. And that is adding actually chloride, the chloride ion, to this peroxide and synthesizing, do you know? A hypochlorite free radical that is essentially bleach. What is the enzyme? This is actually a very important enzyme for you to know. Myeloperoxidase. Just keep that in mind because this is going to be important when you study your immunologic diseases. So, getting back to these free radicals. So we've generated 2 of them. There's another very important one too. This peroxide can actually turn into another type of free radical. It's not physiologic typically. It's generally pathologic. And this free radical is, do you know? The hydroxyl free radical. What do you think is the most common reaction to generate this hydroxyl free radical? It's through the Fenton reaction. And Fenton, the first 2 letters are Fe, so that makes you think of iron, right? It's because it occurs with heavy metals like iron. So actually this iron gets oxidized, okay? Goes from a 2+ to a 3+. And whenever something is oxidized, another thing must be reduced. So what gets reduced? The other reactant, which is the peroxide. Peroxide gets reduced into the hydroxyl free radical, which is very volatile, very dangerous. You don't want that around. You can also have these hydroxyl free radicals formed from radiation. Radiation can split water molecules and actually split off some of these hydroxyl free radicals too. So that's no good. And a final thing that can actually generate all 3 types of these free radicals are just drugs. Whenever some drugs are metabolized by your liver, they go through this phase 1 reaction that can actually synthesize all 3 types of these free radicals and even worse ones that can cause a lot of damage and destruction to tissue. All right, so we talked about how they form and how dangerous they are. Now what? How do we get rid of them, right? That's the main question. That's what I'm asking. So let's start off with superoxide free radical. How do we eliminate this? Well, we actually already mentioned it. What enzyme turns this into this? It's that superoxide dismutase, right? Right. So that will eliminate superoxide and generate more peroxide. But we don't want peroxide around either. So how do we get rid of peroxide? Well, there's 2 main ways. One is the enzyme catalase, and another is glutathione. And they essentially both do the same thing. There's an enzyme that actually will turn glutathione into its oxidized form, and this is called glutathione peroxidase. So it will oxidize glutathione while reducing the peroxide back into oxygen and water. But here's the problem. We actually have to regenerate the glutathione so it can keep working. So what enzyme do you think will react with this oxidized form of glutathione and turn it back into the reduced form? Well, its name will help you out. It's glutathione reductase. We'll regenerate glutathione. We've learned how to eliminate 2 types of these major free radicals. How about this 3rd type, hydroxyl? Well, vitamins is the answer, actually. There's really no better way than the antioxidant vitamins. And the antioxidant vitamins are, here's a helpful mnemonic, ACE. And the most important one for hydroxyls are vitamin C. Now I want to talk about a different type of injury called inhalation injury. So when someone's in a house fire, they're exposed to a lot of smoke. And remember we said the different kinds of injury that can stimulate inflammation. Well, one of them was burns. So actually the incredible temperatures of the smoke can actually burn their trachea. And when you look in their mouth, you might see some edema and swelling of tissue. And if they're having trouble breathing, what are you going to immediately want to do? What's the next best step in management? Intubation. And that's an important thing to know for your board exam. So sometimes when injuries are so deep and so severe, they actually form scars. Scars are essentially, here's an example, you'll all probably have them. They're just essentially connective tissue or collagen at sites where there were severe injuries. So here's a scar. You can see it's contracting the tissue. But sometimes there are disorders with scar formation. We talked about them a little bit earlier, but what do you think this is? So we see that where there was a normal scar, there's actually the connective tissue, the collagen has risen. So it's too much collagen, too much connective tissue. What is that called? That's called a hypertrophic scar, actually. So increased growth of a scar. Another example is this. This is pretty severe. What is this? This is a keloid. And the difference between this and a hypertrophic scar is that the keloid actually grew outside of the boundaries of the wound. So outside the boundaries of the initial wound is a keloid. Within the boundaries, too much scar tissue within the boundaries is a hypertrophic scar. Okay, so we've talked a lot about wound injury, but now we're going to start talking about wound healing. So how does it heal itself? All right, so there are essentially three main phases to wound healing. The first is the inflammatory phase. And so this essentially is right after the injury occurs. Certain cells are called in to this damaged or injured or dying tissue. What is that type of cell? The neutrophils, right? That's right. And why are they called in again? Remember? Well, they release, what do they synthesize? Reactive oxygen species, which go and destroy everything in sight and will deal with whatever pathogen or infection or stimulus is triggering this inflammation. So they're like the cleanup cells. Good. And about when do they enter the tissue? About how long after injury? 24 hours, typically. That's their prime time. Okay, very good. But they also had to get through, they had to burrow holes through the endothelium in the basement membrane, which, by the way, what type of collagen is in the basement membrane? Type 4 is on the floor in the basement. It's a good way to remember it. So they had to burrow holes through the endothelium, and thus we're going to need to patch up these holes. So coagulation will occur during this phase. All right, very good. Then about 2 days later, 2 days after injury, what cell is going to be predominant here? The macrophages, right? Very good. And you can think of macrophages like the orchestrators of everything. They are the bosses. So they're going to tell other cells what to do. And in this case, they're going to tell the neutrophils that, okay, you've done your job. We're proud of you. Thank you for cleaning up. Now you can go. So how are they going to tell these neutrophils to leave tissue? What cytokines will they release? TGF-beta and interleukin-10 are the so-called inhibitory cytokines. So very good. So, and by the way, how did the neutrophils actually leave? Through what process? You remember? Starts with an A. Apoptosis, right? Right, very good. Okay, so that's awesome. Now we've cleaned up everything. The pathogen has been dealt with. Now we can actually start regenerating and repairing tissue. So that's the next phase, is the proliferative phase. It typically occurs approximately 2 to 3 days after the onset of injury, if everything goes right. So the clots will start to dissolve, and a lot of nutrients and also you can think of them as the renovation team will actually have to enter tissue. So where will these new cells and nutrients come from? How will they be transported to this repairing tissue? by capillaries, right? Capillaries, or another name for this is granulation tissue. And what cell do you think actually stimulates the formation of this granulation tissue? That's right, our friends, the macrophages again. So they will release cytokines that stimulate what's called neovascularization. What cytokines stimulate that? Fibroblast growth factor not only stimulates fibroblasts, but also angiogenesis, or this formation of granulation tissue, capillaries. And one more big one, VEGF. Very good. All right, so moving on. We have these capillaries now, and from them, like I said, come the renovation team, which are these spindle-shaped cells. What is the name for these? What are these called? Fibroblasts, right? And what cell do you think stimulates them? The macrophages again, very good. We'll stimulate them to... And what cytokines will stimulate them? TGF-alpha is a big one, and platelet-derived growth factor, good, and TGF-beta. So those are the three main ones you should know. Just commit those to memory, in addition to the other many cytokines you should know. All right, so stimulates the fibroblasts to do what? What do fibroblasts do, actually? They make collagen. What type of collagen this time? There are four main types. What type of collagen is synthesized initially by fibroblasts? Type 3. Very good. Okay, so certain types of fibroblasts can actually contract the injured tissue. What type of fibroblast is that? It's actually the myofibroblasts. Myo means muscle, and so that's because they contract. And so that's actually a very important question on boards. They'll ask you what cell is responsible for wound contraction, the myofibroblasts. So terrific. We've contracted tissue. Now we just need to grow some more cells. So regeneration of the epithelium and other cells will occur, and our tissue will start to look more like it once did. Very good. Now we're ready for the final phase, which is remodeling. So everything's healed. We just need to get rid of the cleanup team now. We're done with them. We don't need them anymore. They will start to disappear about 1 week, so weeks to months later. These cells will start to fade away. Granulation tissue will fade, and so will macrophages and fibroblasts. And then a certain thing happens, actually. This type 3 collagen is converted into another type of collagen. What type is that? Type 1. Very good. So why? Why does that occur? Well, the reason is type 1 is very strong, has the highest tensile strength. And why is that? It's because of cross-linking. So actually these collagen fibrils are cross-linked to one another. How does that happen? Well, something actually places hydroxyl groups onto the fibrils. What does that? What facilitates that? Vitamin C, actually. And then these hydroxyl groups are cross-linked to one another by another enzyme. What is that? Lysyl oxidase is the enzyme. And there's actually a metal that's a cofactor for this enzyme. What is that metal? Copper. It's all very good stuff to know for boards. Now that we have these cross-links, this collagen is much stronger, and therefore this tissue is now much stronger. And by the way, what enzyme actually converted type 3 to type 1 collagen? That enzyme was a metalloproteinase. And there's actually another trace metal responsible for that, and that is zinc. And so while we're at it, let's just quickly review the four main types of collagen. So type 1, we just talked about that, highest tensile strength in skin, tendons, and bones. And that's actually a way you can remember it, is the word bone has the word 1 in it. So that helps you remember that. Type 2, where's that? Type 2, you can remember this because it's cartilage. It's in cartilage. Type 3, we just talked about, it's in the early stages of wound repair. Where else can we see it? It's in very pliable tissues like the uterus and blood vessels. So that's that one. It's very pliable. Okay, type 4, talked about that one already. Type 4 is on the floor. It's in the basement membrane. Very good. Nice little collagen review there.